we try to reach a final diagnosis where you say, okay, which type of malformation you have, and if you eventually look at perform the mutation analysis, which type of mutation.
This week we are talking with the lovely Eleonora Aronica from the University of Amsterdam, who researches epilepsy through neuropathology. Now, for those who aren't overly familiar with the term, a neuropathologist is a person who studies diseases of the nervous system, such as the epilepsies. Eleonora tells us about her research into our brain tissue, which over time improves our understanding of the brain and treatments of the epilepsies. Also, actually it's important, a tip for this week: if you aren't overly familiar with medical terms, do keep the Epilepsy Sparks glossary right next to you, link in the text, as lots of words that many of us are not familiar with are used this week. Stay tuned to learn more, and if you would like to learn more about epilepsy and epilepsy research each week for free, do subscribe to the channel and hit that bell down there for notifications. If you're already actually a fan of the channel, we'd really appreciate it if you could like this video, because it makes a huge difference to us and enables more people to see credible information about epilepsy. And do make sure you check out the links below if you'd like to see the glossary, or if you're interested in learning more about Eleonora and the conference mentioned. Right, as a lemming would say, let's go. Hello, Eleonora. Thank you so much for joining us today. Please, could you tell everybody a bit about yourself?
Thank you for the invitation. It's a pleasure to tell something about myself and the research with focus on epilepsy. I'm originally Italian, you can hear from my accent.
Got that lovely twang, yes.
I moved from Italy in 1991, that was a long time ago. And I started as a student of medicine to be interested in mechanism of brain disease at the time when I was a student. And particularly I did a stage in neuropharmacology where we address also epilepsy. And then I had the possibility when I did my specialization in neurology to do some research. And then I moved for this in the Netherlands, and I thought to stay only 6 months, but then
that's what happens.
I stayed more. I did my PhD in Amsterdam, and I met in Amsterdam a great person and scientist in epilepsy, Professor Lobo da Silva, and thus inspired me to work on epilepsy. But then after my PhD I moved to United States, and I was in Albany and New York. And then I also work on neurological disorder, basic mechanism, including epilepsy. And there I met a neuropathologist, and that was, I thought, oh, maybe, I do neurology another time, a specialization, or I do neuropathology. And then I was ready to start doing neuropathology in the United States, in New York, and then with my husband, he's also a researcher, so I'm married with a Dutch, I have now 2 children, and he's also a scientist working on epilepsy from neurobiology.
Oh, perfect.
So then, but then in in 96 we decided to go back to the Netherlands, so we moved back. I have to restart again, and then I thought, oh, what I have to do? I would like to do epilepsy research and neuropathology, so I wrote my own project, and this was supported by fellowship at the Epilepsy Institute of the north of the Netherlands, SIGN. And thanks to this fellowship, with my own project I started to work at pathology. I did research, but I want to do the second specialization in neuropathology, and was possible. I have to say that neuropathology in the Netherlands is not a separate program, it's a training program, so you have to go through neurology or through pathology. I did through neurology, so I'm a neurologist and registered as a neurology. But I had the possibility to do this specialization in neuropathology with focus on neuropathology of epilepsy, and then I built slowly a research team with a link to SIGN, which is the research expertise center for the north of the Netherlands, and link also to more basic science of the University of Amsterdam for the experimental model. So we try in our team with a young scientist to combine the basic research, so the mechanism of epilepsy, seizure development, progression, pharmacoresistant, with the patients, so with really the clinical work, using human material, so we are in neuropathology based, so we combine both the experiment in models, in vitro, in vivo, and the human tissue obtained at surgery from patients with drug-resistant epilepsy.
So is that where you get the majority of your tissue is from surgeries?
The majority of tissue indeed is from epilepsy surgery, and this is in collaboration with colleagues also at the University of Utrecht, where, for example, more the pediatric surgery for epilepsy is performed. And this is a good characterized tissue. And within our project we characterize also more recently the genetic features of this tissue, looking for specific alteration in the tissue, and also clinically good characterized. So we look at different aspects using the tissue and getting information, data to understand why appunto this tissue is epileptogenic. And then we have also some cases of post-mortem material. They are rare because not a lot of, there is not a biobank which is dedicated to epilepsy as in UK. And we are interested in genetic epilepsy, and most of the time this post-mortem material is not available, so until there is not permission for autopsy, there is not, as I say, a dedicated biobank. This is a pity because the surgical epilepsy allow us to study only a group of disease, but other disease where surgery is not performed, we don't have tissue, so we cannot validate the models.
Oh gosh, that is a real shame. It's like missing a trick, really.
Yes, that is really a pity. I always say, yeah, it is a pity that it's not a biobank. Clearly, if we think a genetic epilepsy, which are not target surgical intervention, it is difficult to get a permission of patients which also are in institutions. So I think it's possible to get to establish a biobank, but you need really a big effort and organization. There are biobanks for all the neurodegenerative disease. We have a great biobank, a Netherlands biobank also now for psychiatry disease, but there is not a biobank specialized focus on genetic type of epilepsy, which is also topic of our focus of our research.
I have to say, just get this in quickly, for those who aren't actually seeing the video, there is this rather la-di-da, rather expensive looking microscope behind you right now, isn't there? Do you actually use that one, or?
I use still the microscope, but we are going to digital pathology, so we have a little bit of delay in our department because of the fusion of the two hospitals, but in other departments they use only the computer, so you get in the place of microscope. I don't think that I will have my microscope in the next 10 years, but the next generation of neuropathology probably they will work with a big screen, so you have 3 screens together, and then you can also do the diagnosis on with digital imaging, which offer a possibility also to analyze the imaging, so to recognize patterns of disease, of a pathology.
Oh my goodness, you're putting like these images into my head like of NASA or something, where you've got like these big screens and you can see all these details in front of you, you know?
So, yeah, actually we'll be as the neuroradiologist for the future. We already use the digital pathology. We are now, for the organization, we are a little bit delayed, but there are, as I mentioned, other hospitals where they only work with digital pathology.
So cool. And so could you tell everybody, please, what led you to the epilepsies? Because there's so like an indefinite number of sort of projects you could, or topics you could go into. Why epilepsy?
Yeah, as I mentioned, I went to epilepsy because I meet some key persons that inspired me to work on epilepsy. And one was when I was a PhD student here at the University of Amsterdam, Professor Lopes da Silva. Really a great and known person in the field of epilepsy and physiology of epilepsy. He also had a clinical, he was a psychiatry for origin, but he did basic research at the University of Amsterdam, and working and developing animal model of epilepsy. So he was really a great person, and he was also the mentor of my husband, which was working on epilepsy. So that was the other important person in my life. So I met as PhD student, and we start to work together. So we start to discuss about the mechanism of epilepsy, the model. When we moved to United States, we continued to work together, and I had also a project at the Albert Einstein College on kinase model of epilepsy and mechanism of epilepsy and glutamate receptor. That was my topic since I started to do research. And when we came back, I had to start from zero because I was following my husband back with a child. So
must be love.
So then I decided, okay, I want to work on epilepsy, and Professor Lobo da Silva stimulated me to write a project, and that I did. I follow his suggestion. There was a fellowship, and I wrote my own project on tumors and epilepsy. But these are special tumors. They are developmental tumor which are highly epileptogenic, which are operated in the context of epilepsy surgery. So my first, my own project, when I really started my small team with one technician, was on this tumor. I'm still working on this tumor, so understanding why these tumors are so epileptogenic, why they are associated with seizure development progression, and this in young patients, so it can be children, young adults. And then I start to work and look at the molecular characteristics of this tumor. 30 years, more than 20 years later, we know more about these tumors, and we know that they are developmental tumor, and so we can include this tumor in the large spectrum of, we can say, developmental disorder, which include also the malformation of cortical development, which is now another topic of my research.
So what have you found out over these past sort of 20 odd years that has sort of, what information or facts have you gathered that has been of use to people affected by the epilepsies?
Yeah, for our research, studying and using the human tissue with different techniques, which include also transcriptomics, so the gene expression, and combining also with experimental model, we, for example, identify inflammation, neuroinflammation of one of the mechanisms which play an important role in seizure development progression, and which represent a commonality throughout a different type of epilepsy. Also there, you don't do the work together, you team together with other colleagues, and great collaborators, particularly in the field of neuroinflammation, is Anna Maria Vezzani. So I think I met her when I still, I was at not a position, that was in 97 at a meeting, and I was also inspired by her presentation and research. And then I think since I met in 97, and since 98 I think we collaborate with her. Still we write paper together, we brainstorm together, and so neuroinflammation was indeed one of the finding which at the starting from 2000 highlight this commonality and potential target of therapy. And then also there I met another person, another meeting, Peter Crino, which was working on developmental disorder and mTOR. And this inspired also me, there is some link between this pathology and this developmental tumor. So I started to work on tuberous sclerosis complex. And this is really one disease that where we work more than 10 years, maybe 15 years within our team. That was thanks to the inspiration that going to the meeting, it's like, oh yeah, that is really an interesting disease to understand the epileptogenesis, the early epileptogenesis, so in children. And then we start to work on the mTOR, this pathway which is associated, which is deregulated in tuberous sclerosis complex, but is involved also in other focal malformation. And in the last 10 years we learned a lot about the genetic of this malformation of cortical development, and this is indeed the one topic of the presentation at the meeting in Denmark. Really the big changes in the understanding the pathogenesis of this developmental disorder is represented by the detection of mutation. Mutation in specific genes which are linked to the mTOR. So there are different genes which regulate with this pathway, which is important for brain development and function. So it's essential to have a normal brain with a normal structure and normal function. And indeed, already before the discovery of these genes, we saw that there were some malformational cortical development which were similar under the microscope. So under the microscope, you could see the same type of alteration, and these are represented by large cells, abnormal cells. There's also inflammation, but particularly what this features is abnormal cells and a disorganization of the structure. So before the genetics, we thought, oh, there has to be a commonality, has to be the same mechanism. And indeed, genes related to the same pathways are involved. And now, as I will discuss also in Denmark at the meeting, we have the possibility to detect this mutation in the tissue. And the challenge is that you cannot detect in biofluid, but these mutations are only present in the tissue, are only present in a small population of cells. So you need really to have tissue, which is good quality tissue.
So you can identify it only in the actual tissue, is that correct?
You can find cells which contain the mutation, okay. And these are structural alterations, so they are malformation of the cortical development, non-neoplastic, but they have a mutation in the pathological cells. And so now we have the tools to detect, because you have to do really deep sequencing to detect this mutation, and 10 years ago we had not the tools to do this. And the idea is now to implement, actually we can implement in the diagnostic workflow, so we can learn, really show that there is a specific mutation in the mTOR pathway, or specific mutation in other pathway. Also other genes not related to mTOR have been identified, and indeed in the update classification of focal cortical dysplasia, the genetics has to be integrated. So the suggestion is really to combine, not only to look at the tissue under the microscope as a classical neuropathology, but to combine this information with the clinical information, also the radiology, and the genetics. And the genetics is really important to have a final diagnosis. So the patients will get a diagnosis, at least we try to reach a final diagnosis where you say, okay, which type of malformation you have, and if you eventually you look at the, perform the mutation analysis, which type of mutation. And this is important, so we hope that will be important to stratify patients, to understand which patient may profit or not from surgical resection, and in the future also which patients may be candidate for a more target therapy. Because if you have a gene, you have a pathway, so you can stratify for a biological category. And just to say that we have inhibitor, for example, of mTOR, which are used in tuberous sclerosis, so that is an example, but there are other possibilities if you identify the gene and the pathway to have a more target therapy. So that is the reason why it's important to have all this information together and to perform a more integrated diagnosis.
And all of this research, especially that which you're just speaking about, tuberous sclerosis, is the information you gather likely to positively impact the care and treatment of people solely with the diagnosis, or do you think it could actually positively impact people with other types of epilepsies as well?
I think the knowledge about the role of mTOR in epileptogenesis is important not only for genetic type of epilepsy, as tuberous sclerosis or focal cortical dysplasia, but for other type of epilepsy. So we know that, for example, activation of mTOR take place also in acquired type of epilepsy. So in temporal lobe epilepsy, and as has been shown in models of temporal lobe epilepsy, there is also an activation of mTOR. So this pathway, which is really complex, is particularly interesting to understand the commonality in epileptogenesis throughout the different type of epilepsy.
That's a really great
look
point.
at the downstream targets. So, yes, so we learned, I think tuberous sclerosis was really, and is still a really great disease model to understand epileptogenesis. And then to translate part of this information to other type of epilepsy is also possible. The advantage of TSC is that you know that the children, they will, or 80% of the children, they will develop seizures. And the children, they have already a network which is pro-epileptogenic before birth. So that is the reason why. So you can follow in the first year of life. So it is really, we can say, a model for epileptogenesis and to eventually modulate epileptogenesis, to modify the severity of disease, because you can follow from the genetic diagnosis at birth and then you try with treatment and early treatment to modify the severity of disease.
Do people with, just making me think of a friend of mine who's mentioned babies or fetuses having seizures. Does that ever happen with tuberous sclerosis?
There are a few papers which support upon to this. What I can say that there is an structural alteration and the functional alteration are already present before birth. So majority of these children develop seizures really in the first months of life. We did, and within the consortium EpiStop, 94 children with diagnosis of tuberous sclerosis at birth have been studied, really with everything, with all the tools that we had in the first 2 years of life. So, for example, doing EEG, the first EEG after the diagnosis, already showing these children alteration of the patterns, of the EEG patterns. And this alteration can be used as a biomarker to predict which children they will have more problem in also in development. So you can use, and this alteration are already present in the first months of life. So that say that we have to start, if we want to modify the severity of disease, as soon as possible with treatment. And in this consortium was done a preventive treatment with vigabatrin, which is one of the anti-seizure medication that you use in these patients. But there are also trials which are going on with for prevention, because that is the idea in this genetic epilepsy, with mTOR inhibitors. So we will hear in the next years more about the results of early treatment prevention.
That's what I was going to ask you actually. What do the next few years hold? So that's what's exciting, I think, is that you carry on with your research and fingers crossed, hopefully, or two fingers crossed, that you will be able to help prevent these seizures and prevent, well, the brain mucking itself up, I guess. That's not a very professional way to say it, but yeah.
Yeah, I think the idea is, and not for myself, but for the clinical colleagues which are involved in clinical trial, is maybe we cannot prevent, but we can modify.
Okay.
And for sure, to modify a process, because the damage, the alteration is, you have to think as epilepsy as a dynamic, and is a network disease, and this network is dynamic. So if you start early with treatment or target treatment, you can control the network. Maybe you cannot prevent seizure, but you can reduce the severity. And what is important that still we could not find in the trial with vigabatrin is eventually to look whether early treatment or target treatment has an impact on the comorbidity. For example, which affect the quality of life of patients, a caregiver. You can control seizure, but if the child still has a cognitive problem, neuropsychiatric disorder, behavior disorder, that is still remain a big problem for the patients, for the child, and for the family.
I'm so happy that you actually mentioned that bit. I actually did a post on social media this morning. No, you don't like social media, but and it's about if anybody's interested, it's a global survey called Epilepsy Surgery, Patient and Carer Perspective. And I did the questionnaire myself this morning, and what was a really, I think, valuable opportunity, and hopefully will be valuable information for the researchers, is that there was a question about, okay, what is your priority when it comes to epilepsy surgery? And because people, I think, generally often think, oh, it's about reducing or getting rid of those seizures. And my input was like, actually, that wasn't my number one. That wasn't my priority. It was about things like mental health, psychiatry. It was about cognitive function, all sorts of things. Like, it was about not being so tired all the time. And so I really appreciate what you were just saying, that it's not about the seizures alone.
No, it is a complex disease with different components, and in genetic epilepsy, for sure, you have two components: epilepsy and development. They are intersecting with each other.
Yeah.
So you can imagine that if you have a genetic epilepsy, you are dealing with mutation of a gene, which is important for normal development. So that itself may affect the development. On the top, you have also epilepsy through the same gene and the mechanism. And if you cannot control seizure activity, then for sure severe seizure activity has also an impact on development. So they are sometimes independent mechanisms related to the genes, but these two different mechanisms, they intersect, they influence each other. And I think that this is, we don't have only to look at control of seizure, but we have to consider in total the problem of the patients. And as mother of a child with intellectual disability, autism, I understand how these aspects are really important and affect the quality of life of the patients, more sometimes of the epilepsy itself.
Thank you, exactly. And thank you for your openness there. I've met many people, I think I've mentioned this in other podcasts, but just, so many people would rather have an extra seizure if it meant that they would be happier, or more content. Or because it's not the priority is not necessarily the controlling or halting of the seizures. And then you have to take into consideration the impacts of potential anti-seizure medications, An interesting one, something I came up with today was that, so we know that, say if we're talking about temporal lobe epilepsy, commonly the impact of both the epilepsy or the sclerosis and the drugs can be reduced cognitive function. And then, okay, so you hope if you have surgery, if you're suitable for surgery and it reduces your number of or severity of seizures, that's great. But you're missing brain tissue and that can severely impact, of course, your cognitive function. So it's kind of weighing things up, isn't it? What is going to be the impact of certain treatments, whether that be surgery, drugs, etc., etc.?
That is the reason why the focus of the research and the treatment, the care for patients with epilepsy, but all the neurological disorder, is to go for a personalized therapy. What is good for one patient is not good for the other, and you have to look at the situation per patient, which is more complicated. So not only seizures, but all the problems which come with seizures and all the side effects of anti-epileptic drugs. And sometimes you do not realize as a neurologist when you give an anti-epileptic drugs how the side effects can be worse. And I have my own experience with my best friends that had a metastasis of a brain tumor, a metastasis of a lung tumor in the brain, and where she had some seizure, but at the end she accepted to have seizures in the place to have anti-seizure medication, because the effect of anti-seizure medication for her were worse than the few seizures that she got per month.
Often it can depend upon a person's, for instance, their profession. I know somebody who has epilepsy but chose to not have a certain type of surgery because she said, I won't be able to paint anymore if that happens, or might be, my level of creativity is likely to be affected, should I say. So no, I won't go for that. And so, yeah, it's different priorities in life, isn't it? I suppose a lucky thing, maybe lucky is not the right word, but if you have this choice when you are older, and you have the intellectual capacity to make that choice, that's really cool, whereas lots of people don't have that.
Yeah, so the parents have to take this decision is, I think, really difficult to decide, but I think with teams and a network of colleagues with different expertise, so I think it's important to go for a personalized therapy and to look at the whole problem of the patients and what is the best for these specific patients. and that is, and communicate with patients, and that
yes, knowing how to do so. Gosh, you are in a remarkably unique, well, kind of on paper sometimes positive, position, that you empathize with what some parents can be going through, the patients can be going through. You've been a neurologist, now you're a neuropathologist. It's seeing all these different aspects is really rare, but hearing from you as well is making me get even more excited about our conference in Denmark, because I know some of the other people there, and I won't mention them because I'll get embarrassed, but just like other, geneticists and stuff, they're really seeing things from the perspective of precision therapy, which is wonderful, rather than just throwing the same anti-seizure medications at everybody and hoping that will be okay, and it's all just about the seizures. Instead, it's about evaluating the lives of each individual with their input, rather than just making the decision as a clinician,
Yeah. That, as we discussed before we started the interview, is really important to talk with patients, to understand what is important for them, and bring this in our research. And that is maybe more straightforward for clinical research, but more difficult if you go to basic research. That is the reason why I try to stimulate young scientists, which are coming from the field of neurobiology, so it's not medical doctor, also to understand how important it is to be confronted with the disease, to understand what they are studying, because they never saw a patient with epilepsy, never talk a patient with epilepsy, but they need to do. They need to do to get inspiration, to understand that they do something which is meaningful, not only looking at cells and doing experiment for their publication. It's not a high impact publication which is important, but it's important if you do things with patients which have an aim, which is to treat the disease, maybe not immediately, but you provide a small information, a small piece of this big puzzle. And then that is one aspect for them to understand the disease, and they do something which is important in a way, but also try to communicate their difficult, complex experiment or results to patients, or maybe help the patients to explain, to discuss a scientific paper of other teams, which could be of interest also.
Yeah. And actually that makes me think of what you just mentioned earlier as well, how you may be rather specifically studying, or one of your areas is tuberous sclerosis, but you also recognize the benefits actually of the data collected for people who aren't affected by tuberous sclerosis, but instead, say, temporal lobe epilepsy. There's a lot of overlap, isn't there?
Yes, we work also in temporal lobe epilepsy.
Oh my God! Okay,
because it's in my heart recently.
Well, that's for another episode. Then we'll go into that one later.
No, actually, I start to work, and we work still on temporal lobe epilepsy, because all the model that we have are model of temporal lobe epilepsy, or post-status epilepticus. So we have two lines of research within the team. One is indeed the genetic epilepsy with focus on enteropathy, so tuberous sclerosis complex. And the other is indeed temporal lobe epilepsy, hippocampal sclerosis, with the model of hippocampal sclerosis, and also intractable status epilepticus. That is also another type of research. What is a commonality indeed, as I mentioned, is the inflammation. The inflammatory pathway that we see in temporal lobe epilepsy, actually we we identify using a model of temporal lobe epilepsy, are also the regulating genetic type of epilepsy, at least in this enteropathy. So there are a lot of commonality. Clearly there are also some different, but there are a lot of commonality. So we try to look at difference, but also commonality, and then to address, for example, the role of mTOR also in temporal lobe epilepsy, the neuroinflammation also in temporal lobe epilepsy, as we do in genetic epilepsy.
Do you know what? I think that's a really good point to end this conversation on, because you just made me think of how us as individuals in this world, or nearly 8 billion of us, we should be communicating with each other, identifying differences, yes, but also identifying similarities and sharing that information to benefit each party. I think that's a really good point to finish on. Thank you so much for your time, Eleonora. This is like so interesting, and I will totally grab you again for, that sounds wrong, but I'll totally, if you are open to it, we will definitely be talking in another episode about your temporal lobe epilepsy research. It's very, very interesting.
With pleasure.